molecules are going to be transported from their higher concentration to their lower concentration, and this is called diffusion. They simply pass the plasma membrane and they come from the higher to the lower concentration. Okay. And they can only diffuse if they are lipid soluble or if they are plasma membrane soluble, right? Now there's a specific term that is used for water only, and this is called osmosis, right? So the plasma membranes, they are partially permeable. Permeable means to allow something, right? So they partially allow the water molecules to pass through them, maybe through these channel proteins. They allow the water molecules to pass through the channel proteins, right? So this partial permeability of the plasma membrane, it helps in the transport of water molecules on both sides, right? Did you get it? This is semi-permeable or partial permeable. So if you have, let's say, on one side of the cell, maybe on the extracellular side, you have the solution form. Solution means there are nutrients, there are gases that are dissolved in water, right? And on the, maybe on the inner side, there's only water, right? Or we can flip that. On the inside, there is the solution, and on the outside, there's water only. And now what will happen here? The water molecules are going to diffuse through this plasma membrane to the opposite side, okay? So they are going to diffuse from their higher concentration to their lower concentration. Here you see the solid molecules, they are in higher concentration while the water molecules are less in number. So now here as well, the water molecules are going from higher to lower concentration, but now it involves this semi-permeable membrane, okay? So in literal terms, osmosis is simply diffusion of water. It is only used for water, and it is simply diffusion of water molecules across a semi-permeable membrane. Whenever it involves a semi-permeable membrane and it is water molecule, this is called osmosis, okay?
Okay.
So now what happens here, if water is present in a closed container, whether it is your vessels, whether it is any bowl, any container you have in your lab, right? So the molecules of water, they are going to exert pressure on the walls of that container, right? They have kinetic energy, they keep on colliding with the walls, okay?
Okay, yeah.
So that collision of water molecules with whatever they are in, whatever container they are in, that collision of water molecule creates a pressure. Fine. So that pressure is called the water potential. Fine.
Water potential.
It's called a water potential. So if you have pure water, it means there are no ions, no charges, no solutes present in water. It's not a solution, it's simply water. Its water potential is going to be 0. Fine. Now, if you add sugar, salt, or any other charged ions on this water, in this pure water, right? Now, the collision between of these water molecules with the walls are going to be affected because of the presence of these solid molecules. Now, they have to interact with these solid molecules as well, right?
Right.
So, this is resulting in the reduction of the water potential, actually. Reducing the collisions between the water molecules. So that is why if you have a solution, its water potential is going to be a negative number. Its water potential is always going to be negative, okay?
Okay, yeah.
So what happens here, that these have higher pressure and these have lower pressure, fine? This have more water molecules, this has now less water molecules. So if you place a semi-permeable membrane here again, what will happen here is that this pressure is going to push water molecules to this side, where there is less pressure of water, right?
Yeah.
The water molecule is always going to flow from its higher water potential to lower water potential. This one's lower water potential, right? So the water is always going to flow from higher water potential to lower water potential. So this is going to move here up until the concentration of water in both sides become equal, right?
Right.
So this is how I show it inside if let's say you have vessels, you have arteries, you have veins, or capillary maybe. So this is what happens inside the body. You have the water molecule, whether outside or whether inside the capillary, and then you have these, let's say, glucose molecules, right? So Semi-permeable membrane or the walls of the arteries or the capillaries, they are not going to allow these glucose molecules to pass through them until there is a carrier or the channel protein, right? However, these water molecules can cross the semi-permeable membrane. So they will always go from their higher concentration, which is actually, in case of if you have pure water, this side would always be having a higher water potential, right? So
Right.
they are always going to move from higher to lower water potential, and this is called the, let's say, movement of this is called osmosis of water, right? So there is
Okay.
going
Yeah.
to be osmosis inside your body as well. The
Mm-hmm.
semi-permeable membranes, whether they are inside the body or whether they are anywhere outside in labs or any place you observe a semi-permeable membrane, it's going to allow the movement of water molecules from higher to lower concentration, and this is osmolarity, right?
Mm-hmm.
Based on this osmolarity, or based on this movement of molecules from higher, how does the water decide whether it has to go from this to this place inside the cell, or how does the cell knows that whether it needs to take in water or whether it needs to take give away the water? So this is based on these terms, which is called isotonicity, hypertonicity, or hypotonicity, right? So this tonicity is basically this thing, right? The
Mm-hmm.
permeability of molecules to pass through. This is called the tonicity, right? So these glucose molecules, they are always going to remain impermeable here. They will not pass through this, right?
Mm-hmm.
So if the water molecule comes here and the water molecules are higher in number as compared to the solutes, this is going to have a different tonicity than if there was no or less water molecule, right? So I'll explain here how this tonicity differs, right? This this tonicity is with reference to two environments. Let me go here. So this is with reference to two environments. Let's say, so I'm drawing this plasma membrane, and these are like, if I talk about external and internal environment, that means this is inside of the cell, right? This is your
Right.
plasma membrane, and this is outside of the cell. It can be anything, fine, and it applies to anything other than cell as well.
Mm-hmm. Okay.
So
Yeah.
if let's say if we are talking about this outside and inside, so if we say that iso means, the literal meanings are iso means same and tonic means strength. So this means that if the number of solvent and solute molecules, they are same in an environment, right? So these are isotonic solutions or isotonic environment, which means, let's say if I say that there are 5 water molecules here, right?
Mm-hmm.
And if I say that there are 3 solute molecules, fine. So this
Yeah.
is the inside of the cell. So what will be its isotonic external environment? Isotonic external environment means that there are 5 water molecules outside and 3 solute molecules outside, right? So that
Right.
means the outside of the cell has same strength as compared to inside, fine. So any
Fine.
solution which is present here, any solution which is present here is actually isotonic to the environment of the cell, which means it has same strength as that of the cell. It has same number of water molecules and solute molecules as compared to the cell. Okay,
Cell. Okay. Yeah.
so this is isotonic condition, right? Now this is hypertonic condition, which is hyper means more and tonic means strength, right? If in
Right.
any side, or if on any side the number of solutes increases and the number of solvents decreases, okay, fine.
Mm-hmm.
So if I
Fine.
say that, let's remove this from here. Now there are four solute molecules, um fine. So there you have more solutes outside and less more solutes and less solvent. Let me do some this thing. This means that outside of the cell is going to have more solutes as compared to, if we stick to the to only the number of solutes. So outside is going to have, or maybe inside is going to have more solutes as compared to outside, right? You see more solutes and less solvent. Fine. So now you see here. Wait. Now this portion you see here, you have 3 solute molecules as compared to 2 solute molecules outside, right?
Right.
And you have 4 water molecules as compared to 5 molecules here, right? So this means it has more solutes and less water, and it has more water and less solutes. So and this solution inside the cell is going to be hypertonic, means it has more strength of solutes as compared to this one. This is going to be hypertonic. This one is going to be hypertonic, okay, as compared to this one. Fine?
Fine.
So this is hypertonic, which means that the number of solutes is here. So now there is a rule, I'll explain it in a while. Fine? How this rule works. Now, if we talk about hypotonic solutions, the hypotonic solution means that the strength of the solutes is going to be less as compared to the solvents. Fine, there will be fewer solutes and more solvents, right?
Right.
Now, if this was hypotonic solution, the cell was hypotonic, hypertonic, this is going to be hypotonic as compared to this one. Fine, so this is hypotonic. It means it has lesser solutes and more solvent. Fine?
Fine.
Fine. So what I said earlier was that water molecules are always going to travel from their higher water potential or from their higher concentration to lower concentration. So in this case, what is going to happen is that the water is going to flow from the hypotonic solution to the hypertonic solution. Okay? Because
Right.
it has
Yeah.
more water molecules. So every time you have a hypotonic solution outside, the water is going to flow from outside to inside, right?
You're right.
Is it confusing, or is you have all the concept cleared?
It's clear right now.
Okay.
So
Okay. Okay. So based on this, let's say hypertonicity or hypotonicity, there are two terms which I think I should explain. This is hydrostatic pressure and osmotic pressure, right? So hydrostatic pressure is any pressure of water which is in a closed container. Any the pressure that is created because of the water being in a closed container. So let's say if this is a capillary or a blood vessel and we have water here, any pressure that it will exert on the walls of the capillary is going to be called hydrostatic pressure. Fine? Because of this hydrostatic pressure, water is going to be pushed outside of this capillary, right? So you see, hydrostatic pressure is water being pushed out by some force. If there is a lot of water in the blood vessel, it will get pushed out, causing edema in the tissues. Edema is actually the accumulation of water around your capillaries or inside the cavity. Okay, fine. Now, osmotic pressure is osmotic pressure is simply the pressure of water moving from its higher concentration to lower concentration. So if the outside of the cell, it has more water concentration, so the water is going to move from outside to inside. Osmotic pressure is water moving from its area of higher concentration to area of lower concentration. If there are too many particles in the plasma, water will be sucked into the blood vessel, causing the blood pressure to elevate. This means that if this is hypertonic means it has more solutes and less water, right? This thing. So water is going to be pushed inside this blood vessel because of its higher concentration outside. So this is going to elevate the blood pressure as well. Okay.
Okay.
So this is how hydrostatic and osmotic pressures work in case of hypertonic, hypotonic, and isotonic solutions. So based on this, we have this thing to understand that in case of animals and plant cells, this osmosis or this hydrostatic pressure is creating very important roles. Has very important roles, and it is helping in maintaining the structures of these cells. So we'll understand this one by one. Fine.
Right.
Okay.
Fine. Yeah.
Starting from this isotonic tonic condition, equal water concentrations inside and outside. That means this cell and its external environment are in the isotonic state, right? They have same strength outside and inside. Fine. equal concentrations of solutes and so on. So there will be equal transport of water inside and outside. Let's say there are 5 molecules going inside, so 5 molecules are going to go inside outside, and the pressure of the water on these walls is going to remain, you say, in a balanced. It's going to remain balanced, and the shape is going to remain normal. Fine. Shape of the plant cell is going to remain normal. Fine.
Yeah, that's
However,
fine.
if this plant cell is in such a state that the concentration of water outside is less as compared to inside, which means that the outside environment is hypertonic. Fine. Hypertonic means it has more solutes, less water. Fine. Concentrated solution outside. Concentrated solution means more solutes outside. So if there are more solutes outside and less water, water is going to move out of the cell. Simply water is going to move out of the cell. For example, there's very hot weather and all the water that was in on the outside of a plant cell has evaporated, or that is maybe in the surroundings of a plant, it has evaporated. Fine. So
Fine.
what will happen now? The plant cells are going to release their water molecules into the surrounding to make the concentrations equal. It will try to make the concentration. So it will release all its water molecule outside, and this condition is called plasmolysis. That means because of this release, the cell is going to lose its shape. Fine.
Fine.
So it's going to turn, you can say it is going to become flaccid, or it's going to change its state. Fine. Now,
Fine.
on the other hand, if you have like higher water concentration outside, means that there is a lot of water near the tree or the plant. Fine. So what will happen here? The solutes inside are more as compared to solutes in the water outside. Fine. So the water is going to enter into the plant cell. Fine.
Fine.
If you water too much, if you give too much water to the plant, it will have a higher concentration of water outside. So the water is going to enter into the plant cell continuously, and it will make the shape rigid or turgid. It will put a lot of pressure on the plant cell, right?
Yes.
Did you understand?
Yes.
Okay. So because of this hypertonic condition outside, the water is going to move inside the cell. Now, this is what is happening when you have a lot of water going inside the plant cell. So what happens here? The vacuole in the plant cell is going to store all the water that is coming from outside, and it is going to put a lot of pressure on the cell wall. Fine. This pressure is
Yes.
specifically called the Turgor pressure or the Turgor, right? So this Turgor pressure is actually helping in maintaining the shape of this plant cell. So if up to a certain limit, this pressure can maintain the shape of the plant cell, but if this pressure increased after a certain limit, this might burst. Okay. So that is why there is this cell wall outside plant cells only and not in animal cells, because the plants cannot control the intake of water themselves. We can control the intake of water, but plants do not control the intake of water. They have to take in the extra water if it is present outside. If you water too much, plant has no option other than taking in all the water. Fine.
Fine.
So it will put a lot of Turgor pressure on the cell wall. So this is why you see these two conditions of the plants sometimes. If that is, if like you have a condition that like it's hot, it's summers, or maybe you don't water the plants much. So what is going to happen is that outside of the plant is going to become hyperosmotic, hypertonic. That means it is going to lose its water molecule, and the plants is going to give its water molecule to the surrounding, and it will lose its shape just like this. It is going to wilt. Fine.
Yes.
On the other hand, if you are regularly watering the plant, it is going to stay fresh like this. This freshness is because of this Turgor pressure. It is keeping the leaves erect. It is keeping the plants stand tall. This is happening inside the vacuole if you are accurately watering your plants. It is becoming turgid, it is maintaining its certain shape. But if you don't water the plants regularly, the vacuole is going to lose all the water molecules and plants are going to wilt. Fine.
Fine.
This is how osmosis is helping the plants in maintaining their shapes, maintaining the plant cell, the shape of the plant cells. Okay,
Fine. Yeah.
you get it?
Yes.
Do you have any specific questions related to this topic?
No, not yet.
Okay, so same is the case with, just give me a second. So same is the case with animal cells. In our body, even in blood cells or in blood vessels, what happens is that if your capillaries, they have this condition that the outside of the cell, you have the capillary and there are multiple cells in the blood vessel or capillaries, right? So if, let's say this is a red blood cell, and if in the plasma or in the blood, there is the water condition concentration or the amount of water is equal to that of inside the cell will retain its shape, right?
Mm-hmm.
This is red blood cell, it is disc shape, right? So there is going to be equal intake and efflux of the water, intake and release of water. Fine, so the shape is going to maintain. However, if there is a condition that the blood loses its water, right? You don't drink enough water, let's say. If you don't drink enough water, you are not going to have enough water in the blood. So what will happen there, that the cells are going to lose their water into the blood, and they are going to lose their shapes, so they become shriveled like this. This red blood cell has actually lost its shape if you are dehydrated. Got it?
Got it.
But on the other hand, if the solution inside the blood, or if the blood has like hypotonic solution, it means that there are more water molecules than those in the red blood cell. It is going to burst your cell. All the water in your blood is going to enter the red blood cells or maybe other cells, and it is going to burst these cells. Fine. They are going to lose their shape as well. Fine. So there's a limit of, let's say, intake of water as well. Fine. So this is actually lysed, means this is actually burst because there is no cell wall to retain the pressure, to resist the pressure. Okay, so they are going to easily burst. Fine.
Yeah.
So if you put your let's say if you take out some blood from your body and put it in a lot of pure water, right? You will see after sometimes all the red blood cells are going to burst.
Fine. Yeah.
Now, what is happening in this situation? This situation is called cytolysis. Cyto means cell, lysis means breakdown. Breakdown of cells. So this is called cytolysis, breakdown of cells. So what is happening here? The water molecules are entering inside the cytoplasm. Fine. They are increasing the size of the cell. So up to a certain state, it can increase its size, but because of the absence of cell wall, there will come a stage when this is not going to bear all the pressure from the water.
Okay.
This
Yeah.
plasma membrane is going to burst simply.
Oh, okay.
Okay. So if this is putting, saw that in case of plants, there was turgor pressure and the cell wall was resisting that turgor pressure. But in case of animals, there is no cell wall. So all the pressure from the water inside, there is no vacuole to store it. All the water is inside the cytoplasm. So all this pressure is going to burst this plasma membrane. So you get this burst cell. This is called lysate. Is actually anything that comes out of a burst cell. Fine. So membrane is going to break down, and all these contents of the cell are going to come outside. So these are three stages. Water molecule moves into the cell by osmosis. The cell volume is going to increase up to a certain point. So the cell is basically elastic. It has certain capacity of intaking water, but after a certain limit, it is going to burst.
Yeah.
You see balloons, if you blow a balloon, it blows up to a certain limit, but if you blow it further, it is going to burst simply because you have no protection outside it, right?
Yeah.
So this is what is happening inside the animal cell as well. So this was all about today's chapter. We have